Okay, welcome everyone to the last IGAC ICACGP ECR seminar for 2025. I'm Simone, I'm the co-chair of the ECR SSC, and I will be your host today. We will start out with Shahina Saikh from the BOSI Institute, who will talk about pathogens, airborne pathogens over Delhi. If you have any questions during the talk, please put them in the chat or raise your hand after the talk and we can take them then. Shahina, the floor is yours.
Thank you for the invite. Okay, let me share my screen. Okay. Very good afternoon, everyone. Today I'm here to present my talk that is Invisible Threats Above Urban Landscape, Characterizing Airborne Pathogens in Delhi, India. And first of all, I'd like to give a brief introduction about how these pathogens are in the atmosphere. Sorry, my screen is not working. Okay. Usually, they are derived from various sources, which are continental, sea, industries, and are emitted into the atmosphere. The bioaerosols are then transported over long-range distances when they are attached with other aerosol particles. And they further help in the enhancement of cloud microorganisms and are transported over long-range distances. And due to precipitation, they are deposited into the new downwind regions, which alters the biodiversity of microbial population in the downwind regions. On the global scale, on the global scale, I'd like to say that in the last 3 decades, it's the number of articles published with the keyword bioaerosol is only 3,000. And after USA and China, India is the third population with about 200 research articles on bioaerosols. So it is an exponentially growing field of research. The question, as it is a general audience, I'd like to give a few terms which would further help to understand this field of research. That is genera. A genera is usually used for a species, a living species, where we classify them on the basis of their different classifications, like kingdom, phylum, class, order, family, and genus. And next, I would like to explain operational taxonomic units. Here we have classified the different DNA samples and clustered them together on the basis of similarity. So the DNA sequences, which are 97% similar, are clustered into one OTU sequence. And we have considered that as one genera. Replication is generally the process of further amplifications of our bacterial species. Next is BacDive. It is a bacterial diversity metadatabase where we can get the information of all the bacteria in the atmosphere, or which generally about 92,000 species of bacteria are present here. And bacterial diversity measures are measured with the help of Shannon and Cowan, and it is based on the entropy measurements. So the research questions that we were about to answer here are how humans inherit thousands of microbes, and they are also pathogenic in natures. And due to climate change, urbanizations, and extreme weather conditions, how these bioaerosols function, what are the microbes that we are exposed to in the atmosphere, how the meteorological factors help to determine its communities, and what are the health implications they have, and does human population have any effect on the airborne bacterial populations. The methodology we used for this were we collected the airborne samples from the atmosphere with the help of air sample collection pumps. Then we have stored it in the buffer, which is 3-CDDA, as it prevents denaturation of DNA. Then we have isolated the DNA samples from these samples, and then we performed amplification with the help of polymerase chain reactions. And next generation sequencing was performed to amplify these DNA sequences, and with the help of bioinformatics tools, we assigned the operational taxonomic units. To identify the pathogens, we have used 16S pathogen identification. So we have used 16S pathogen identification pathway, and Here we have deposited our sequences, and then it is a database where about 15,000 pathogen sequences are already uploaded. So they compare the sequences with the sequence that are present in our samples, and then we get the pathogens that are present in our air samples. So the sampling site that we chose for this study was Delhi, which is one of the most popular institutions in India. Within Delhi, we have selected two sampling sites, that is IITM, which is Indian Institute of Tropical Meteorology, and so the second was JNU, Jawaharlal Nehru University. IITM is comparatively more populated than JNU. So the samples were collected in 4 different phases. The first phase we have named it as typical winter conditions. In winters, usually the Indo-Gangetic Plain. Here, we see a high density of fog. In the first conditions where we have collected our samples, a slight depression was noticed over Delhi. So we considered these conditions as typical winter days. The second phase of sample collection was in January itself, and we found a continuous high density of fog throughout Indo-Gangetic Plain. We have considered these as hazy conditions. The third was rainy conditions, where we can see cloud presence throughout Indo-Gangetic Plain. And the fourth is a clear conditions where nothing is a clear sky is visible in the atmosphere of Delhi. So these are the images from the sampling sites during our sample collection phase, where we can see a typical winter day is hazy, but comparatively lesser than hazy conditions. Here, the rain even has occurred, and in seasonal transition conditions, where we have transition condition from winter to summer, a comparatively sunny sky is present. The highest loading was observed in seasonal transition conditions, followed by haze, typical winter, and rain. Now coming to the results of operational taxonomic units, genus, cell counts, and pathogenic OTUs, we found a similar trend in most of the conditions, where we have found highest loading in hazy conditions, a decline in rainy conditions, and then an increase again in seasonal conditions. The possible reason for this is during hazy conditions, high moisture content in the atmosphere and high particulate matters also further enhance the microbial growth, as it provides nutrients as well as moisture helps in the enhancement of bacterial populations in the atmosphere. During rain, as it a washout event has occurred, so all the particles deposited into the ground, and so the population was comparatively declined. And again, in seasonal transition conditions, an optimum condition is again attained as the temperature has increased, and further optimal condition for bacterial populations growth is about 37 degrees Celsius. So the population has again increased during these conditions. So this is plot shows the diversity, how diverse our population was during our sample collection phase. We found there's the highest diversity is in haze, followed by seasonal transition, typical winter and rainy event. And in the second plot, where we have plotted the principal component analysis plots, it is a beta diversity plot. We found a distinct diversity in all the 4 conditions, as the bacterial populations were not similar in these conditions, and different conditions and different bacterial populations. Coming to the Venn diagram, we found that about 10% of the bacterial population were independent of temporal and spatial distributions. So we considered these bacterial populations as the background of atmosphere of Delhi. And later, to see the effect of pollution and meteorology, we found that about pollution contributes about 14 to 20% in the enhancement of bacterial populations, and meteorology controls about 60%, 40 to 58%, which is about almost 60% of bacterial populations. Now, to see the dependency, how they are dependent on different meteorological parameters, we performed a correlation analysis plot, where we found a positive correlation with PM 2.5, AQI, and temperature in hazy conditions. However, a negative correlation was found with wind speed, wind direction, and visibility. So higher PM 2.5 help in the enhancement of bacterial population in the atmosphere, which was further confirmed with CCA plot, where we found dominant bacterial species were highly correlated with PM 2.5 and temperature. During rainy conditions, we found a negative correlation with PM 2.5 and AQI. RH, wind speed, wind direction also showed negative correlation. However, temperature and visibility has positive correlation, as increase in temperature helps in the optimal condition for bacterial growth and enhancement. So temperature and visibility, here we can see that the dominant bacterial species were positively correlated with these 2 parameters. In seasonal transition conditions, we found temperature and visibility showing positive correlations and RH and wind speed showing negative correlations, which was further supported by the CCA plot. Now, coming to the pathogens that were present in our samples, we have found these pathogens with the help of 16S PIP, where different pathogens dominate in different conditions. Like we can see this bacteria, Tapidisphaera, is highly abundant in hazy conditions. However, Deinococcus is present in high abundance in seasonal transitions, similarly for both the conditions. And these were pathogens were further confirmed with the help of literature survey, where we found that all these bacteria were also pathogenic in nature. And on the basis of their target organs, we have classified them into skin, respiratory, gastrointestinal tract, and oral infections. And we found that it is an increasing trend in both IITM, which is high populated region, and low populated regions. We found that during rain and seasonal transition conditions, the intensity and the abundance of pathogens were significantly high. To compare our study with the studies that were performed in different parts of the world, we have downloaded and analyzed the data of next generation sequencing from different parts of the different countries. And we found that Delhi somehow was one of the leading countries with high diversity of bacteria. And then when we performed the beta diversity plot to see how distinct these populations was, we found a completely distinct cluster of bacteria for Delhi's population, which is pathogenic in nature. And compared to the different parameters like OTUs, genera, and pathogens, we found that somehow Delhi was one of the leading countries with high populations of different genera and which are also pathogenic in nature. The conclusions that we came up with: highest bacterial diversity was in haze, and followed by seasonal transition, typical winter and rain. And the bacteria's diversity was completely distinct in all these conditions. About 10% of the bacterial populations were present throughout the sampling phase and independent of spatial and temporal variations. And 20% enhancement was due to population over high populated regions. Meteorology contributed about 60% and air pollution contributed about 20%. And PM 2.5, AQI and temperature showed positive correlation. Wind speed and wind direction showed negative correlations. Human pathogens affecting various organs were abundant over Delhi. In Asia, Delhi somehow has the distinct bacterial diversity and which needs to be investigated further. This shows a summary figure of our study, where we can say that meteorology contributes both positively and negatively to airborne bacterial loadings, and it controls about 60% of the enhancement or decline. However, population and pollution parameters contributes about 20% each, and it helps in the enhancement of bacterial populations. These results were published in the journal Atmospheric Environment, and it is also published in the Department of Science and Technology from the Government of India. Further, it was picked up by different news channels and present throughout the nationally and internationally in different platforms, that bacterial enhancement is related to the concentration of PM 2.5 and pollution. Lastly, I'd like to acknowledge a bunch of people here present, which helped me through this study. Thank you.
Thank you, Shahina. If anybody have any questions for this talk, please put them in the chat or raise your hand and you can ask them yourself. Oh, so Sachin has a question. What is the dependence of the microorganisms number on the relative humidity of the place in general? Is it strongly seasonal?
Yes, due to relative humidity, the population of bacteria are generally increasing as high humidity is present. So bacteria required water content for their growth. So we found positive correlation in general. During foggy conditions, it is a generally increasing trend.
Are there more questions for Shahina's talk? So I was wondering, when you have, like, when metrology is such a big part of controlling whether these pathogens are going up or down, like, there's not a lot of places, I assume, where we can like target to try and minimize it. Or am I misunderstanding that? Because we can't really do that much about metrology.
Yes, we can't control the meteorology, but we can then take suitable precautions for particular type of pathogens that are increasing, like skin, respiratory, and different. We can take precautions for that. So we need a long-term data for this. It is just a 2-year data. So we need long-term survey so that we can then plan it accordingly, like what type of pathogens are dominating in which seasons.
And with the like pollution being 20%. So if we
Yeah,
reduce the pollution, we will also be reducing the pathogens
20%. Yeah.
20%. And you know that is it a linear correlation or is it like so if you reduce the pollution by 10%, do you also reduce the 20% impact that's coming from pollution by 10% or
It's a linear correlation because the bacteria need something to survive in the atmosphere. So they are somehow attached to a PM 2.5 particles because the sulfate, nitrate, there are different types of bacteria. Some are sulfate loving, some are nitrate loving bacteria. So if we reduce the PM 2.5 concentrations, it will automatically help in reducing the bacterial population as well.
because then the bacteria would go to the ground
Yeah.
instead of
Yes.
being in the air. Okay.
Yeah.
Are there any other questions? Okay. Then thank you, Shahina. Then we
Okay.
will move on to our second talk of the day, which will be by Merve Polat from the University of Copenhagen and the National Institute of Working Environment in Copenhagen. And she'll be talking about emissions from asphalt.
Okay, it just you can see my screen, right? Yeah.
Yes, but not your slides.
Yes, I'm just gonna start them now. Just
Perfect.
gonna check that I chose the right slide because somehow I have two on. Okay, everything good?
Yes, that
Yes.
looks good.
Okay, yeah. Thank you for the invite. So my name is Merve Polat, and I'm from the National Research Center for the Work Environment and University of Copenhagen, Department of Chemistry. And I'm going to talk a little bit about my PhD project. So I'm a third-year PhD student, and this is still an ongoing study. So I titled this one "Paving the Road Between Field and Laboratory Studies." So my PhD project in itself is titled "Occupational Environmental Implications of Greener Asphalt Alternatives," and it's, as the name states, to investigate emissions from different types of asphalt. And the reason for that is that when we move against a world where we want to reduce our carbon footprint, we have to also know what kind of new material we introduce, like the new type of fumes and things we introduce to the environment when we are changing the material we use as well. But the thing we're going to talk about here is most related to the asphalt. So I'm going to take the green stuff out, but this is kind of the scope of my project. And what is asphalt really, you might ask? So a lot of people, they typically say that asphalt is roads, cars, highways, such things. I learned through my study that when you talk about asphalt, you talk about two things. You talk about the binder, so like what is inside, like what makes it kind of black, gray, and so on. This is typically referred to as bitumen. And then this is, so bitumen is made from when you refine oil, then you have like a byproduct left. This residue is what you put into the ground. And then you have the filler, which is all the stones and so on that make up the biggest part of the mass. And before we can use this asphalt for the roads, we have to produce it. And then we have to construct the road as well. The construction of the road typically happens in 2 stages. So you have the milling, take off the old layer, and then pave, you put on a new layer. So the cycle of asphalt is then you produce it, use it, lay it, use it, and then you take it off and throw it out. That's the main process. Then this process is then when you construct the road, you mill it first, and then you pave it. So you might ask, what can an atmospheric scientist or chemist say about asphalt? So the things I'm looking at is when we construct the road or we use asphalt, how does it affect the workers that work with it? So the fumes, and how does it affect the general public but also what kind of impact can it have on urban air quality. And the way we are doing this is looking into this exposure pathways that we identify in my thesis. So for example, you have some asphalt right here. You have a lot of these organic vapors. A lot of you might have already smelled, like when you smell asphalt, it has a distinct smell from like as quite rich in organic fumes. And what does these organic fumes contain then? So there have been studies where they have looked a lot into, especially in the laboratory, where they've looked into, okay, what does it contain? And they see, okay, it contains a lot of semi-volatiles, pHs, especially like different sulfur and nitrogen aromatics and so on. And one thing they also see is that the emissions, so like how much is emitted from these samples, increase with higher temperature. So in the field studies that they have looked into that asphalt workers are exposed to higher elevated numbers of particles from bitumen compared to the background, but it's still kind of unknown how other pollutants in the air affect these bitumen fumes, especially the exposure to them. So what we are trying to investigate is how well does these laboratory emission represent real life scenarios? What is the chemical composition of the asphalt fumes then? And how does it actually change when you then use green asphalt types? And how does it then affect that you introduce other things to that matrix, such as soot and so on, that are present in roads around the world? So we characterize two main pathways. One of them is this one, which is called POA, so primary organic aerosols. And what it is that, let's say you have some seeds present. This can, for example, be soot or other things, ammonium sulfate, ammonium nitrate particles that are already present in the air. What happens is that these condensate will condense on them. You start forming a surface on them. This will lead to exposure in a shorter time range. So this will affect the workers and the general public, more the workers. And then the second one is then SOA formation, so secondary organic aerosols formation. What happens is that these organics are oxidized by oxygen in the air. Then you start forming low volatile compounds that can start nucleating and forming new particles. This will happen typically on a little longer time scale, so this will affect both groups. So the implications for these can also have implications then for the climate, for cloud formation and so on, but this is not something we specifically look into. So how do we tackle these problems? So as you might have guessed, we combine these field study together with laboratory studies. And I'm gonna show an example here of a field campaign. So this is a field campaign I did together with Lund University. And as you can see up here, can you see my cursor? I guess, yes.
Yes.
So this is a field campaign I did together with Lund University. And then we were looking into milling and paving sites in certain part of Sweden. And what milling is again. So milling is when you have old asphalt layer, you strip it off and then you sweep it afterwards. And then it's ready for the next day that you then have a asphalt truck coming with hot asphalt, and then you pave it, and then you compress the asphalt by these rollers. And the way we have done these measurements is by doing personal exposure, and then this little station we have here. So we let all these things pass by, and then we get like these episodic snapshots of what the fumes look like. And then another thing we have done is these small backpacks you can see on these machines right here. And the idea with them is that we get more source-specific compounds, or we can look at those source-specific rather than with the workers. They typically move around the different sources we have thought about there might be. And then we're like, okay, we have to have something that are more source-specific in that sense. And the offline analysis, what we do is we look for different type of pHs. They can be like normal pHs, and then alkylated and sulfur containing pHs. We look at elemental carbon, organic carbon as well. And then for the real-time analysis, we we get a lot of data, and then we have a lot of instruments, and then we analyze that. So some of the results might look like this, but I don't think you will get that much out of this. So I try to visualize in a different way. So when we then look at how the concentration changes. So for example, look over here. This is the background of the milling site. And for the people who are not in aerosol science, PM1 means particulate matter with for particles that have a diameter less than 1 micrometer. So it's the mass of those. And then PM10 means the ones that are from 4 to 10 then in this plot here. So one thing I want you guys to notice then is this is the background what we start out with. When we start milling, this increases right here. So we have much more coarse particles, so we have big particles. And then when we're sweeping, we have even bigger particles. When we then look at the paving side, we have a similar background. So we have a different background. But what happens is when we start paving, this PM1 increases. So these are like the small particles, PM1 particles. And you see them increasing, and then you have a little less here. And if you look at the numbers here, this is quite high for small particles. Another way to look at this is what we call PN, which instead of looking at the mass of the particles, we then look at the number of the particles. And so this is sub-micrometer particles, so it just defines a small particle sizes. And what we see again here is that the paving machine, the roller that compresses asphalt, and then the milling machine all have quite high levels of these really small particles. And then when we look at the chemical composition of these fumes, we see that for the milling, we have a lot of combustion particles, big particles. We have what we call high molecular weight pHs. And then for the paving, we have these more low to middle molecular weight pHs, much more organic particles, finer, and then also some that contain sulfur pHs and alkylate pHs. And then one interesting thing we also saw was that when we look at these source-specific sources, we see, okay, this milling machine has similar profiles to what we see for the paving day. So what the conclusion for this part of my presentation is that asphalt fumes, they are comprised of fine organic rich particles that are enriched in these different type of pHs. And then we did exposure ranking, so paving and then compacting by the roller and then milling are the ones where we can see there's asphalt fumes. And then we were like, okay, we also did a personal exposure study parallel to this one for the same sites. And what we see is when we have the workers that go around the different type of sources, it's hard to figure out what exactly activity that the workers do can actually contribute to that exposure. So by doing these source-specific ones, we can actually better understand this process leads to some kind of exposure. But there's always some problems in the field. And that's why I'm also doing a lot of laboratory work where I look more into these processes. Example is here. So this is where we look into these PUA particles. So for example, number one here, you have the aerosol seeds, you have the asphalt condensate, they go through a flow tube, and then you start forming these PUA particles. And what we measure with is an AMS and ACSM. This measures the chemical composition of the aerosols. And then SMPS measures the size and the concentration. And this one measures the absorbance of the aerosols. This is what it looks like. It's not that pretty. And I just took a course where it was told to be very transparent. So I'm going to briefly recap 9 months of trying to get this set up to work. So one thing is that this is the more simplified version of the setup. So we heat up the asphalt, we go through the flow tube. So one thing we had a problem with is that one thing worked and then there was a problem. So one thing was that if you, for example, have these seeds, which is this yellow one, we start heating up the sample. These dots right here is the temperature. And the organics you can see right here. So we start forming this core around. So this one is growing into these bigger size particles, but then this blue one also starts increasing. And this represents self-nucleation. So where these condensate from asphalt, they nucleate themselves rather than actually sticking on the surface of the seed aerosols, which was an uphill battle for me to design my setup. And so what I did, I tried to change the seeds, didn't help that much. I tried to make a better heating line through and more heating, and it kind of worked better and better. So what you see here is that these is like you want them to go to dark blue right here. So by doing alterations, it got better and better, and we had a lot of losses. It also slowly got better by neutralizing the particles beforehand, and then at last I got it to work. And what you see here is that this red and orange ones, these are the seed particles, ammonium sulfate I'm using here, and the organics are then the green ones. And what we see here is when we increase temperature, which are these dots, we start seeing the organics coating. And what we also see is this mode, we start here. With asphalt temperature, this mode of aerosols, they grow to bigger sizes, which again shows that we have this condensation happening. And then another way to look at it is to look at the mass spectrum for these different periods that I have marked right here. So another way we look at it is by the organic fraction. So this is how much this green one makes up of all the other ones. And then what we see is we only have seeds. This is what the max spectrum looks like. But then when we add the asphalt, we start seeing more and more these organics, so the green peaks right here. So these are, for example, this is number 3 right here, and then this is number 4. So I was very happy because that meant that I could go to Lund University and then use some of their instruments. And this is what I did. And this is what it looks like now, the setup. So I have introduced 2 new instruments, and I have redesigned the setup, and I'm working there. So these are quite new data from 2 weeks ago. But similarly, what we see is that these different periods, I have then marked, for example, there are different temperature settings, or they're like on and off seed settings. So what we see is that if we only have the seeds, this is what the mass spectrum look like. And then when we put in the asphalt, we start seeing some of these organics. So these like organic peaks. And then when we increase the temperature, it becomes more and more apparent. So again, another way to look at this is the organic fraction. We see it making up here. So when we have the seeds, we have typically CH family groups like this. Or we have like, there will always be organic trace with the AMS. It's hard to get away because it's very, very sensitive. But what we see when we have the seed is like very low concentration of the organics, so almost nothing. And then we go with this number 3 where we add the asphalt sample, and then we start forming this coating where we get more of these CH groups. And then number 4 is when I take away the seeds, so there should be nothing happening, which is ensuring that there's nothing. And what happens is when we then increase the temperature, we start forming, as you can see, more of these CH groups. But what happens then when we increase the temperature even more is that we start seeing that these 2 increase. So the red and the yellow one, which are these oxidized carbon families. And this is quite interesting because then when you look at a Van Kievelen plot, where you can look at the hydrogen to carbon ratio as a function of the oxygen to carbon ratio, what you see is that when we start increasing the temperature of the asphalt, so different operational temperatures, then we start seeing that we start emitting more oxidized compounds. So this is what we see. We go from a low oxidation state to a higher one. So this is quite interesting. And the next stage for this type, this one will then be to test different types of asphalt and see if they have similar processes. So if we then go to the field, how does it look like? We took out a car with our instruments and then read out and measured. And these are 3 different roads in Copenhagen. And what you can see is that when we have the paving again, we start seeing elevated levels or like elevated levels of organics, but also the mass spectrum that changes, similar with compacting. And then if we look at how the second pathway are defined in the beginning, so the SOA pathway, what I did was I was at Cork University, where I work with Amir Bendrik, and we looked at simple systems. We know we have a lot of naphthalene, we have a lot of benzene that co-react with the naphthalene and can affect the SOA formation potential. And then we're looking at how that affected by other gases that are present in roads. And this, for example, NOx and SOx. So we looked at 8 different reaction mixtures. And then, for example, naphthalene plus OH is reaction mixture 1. Naphthalene OH plus NO is reaction mixture 2. And what we see is that NO actually accelerates the SOA formation. So we form particles much faster. And we see that benzene delays this SOA formation. So that's why it's really important to include different co-reactants to your mechanism, especially when you have environments such as asphalt emissions, where you have a lot of other things that also are present. And one other thing we also saw that NO did was, if you look at these colors up here, these are different oxidation states of the gas phase. We see that we actually start this color scale. The schedule is different than this one over here. So reaction mixture number 2 and 5 have NO in it. So what we see is that we form these higher oxidized species much faster with NO than without. And then the next thing I'm going to do at Lund is use the authentic samples and then see if I can get similar things, or kind of combine it to other things I had. So did we manage to pave the road? I was just going to say not fully, but we're on our way. And the thing that I want you guys to take away from this talk is that asphalt is quite fun because it has a lot of, it's a big cocktail of a lot of things. So you get to try different methods and different way of thinking. But the more scientific things you should take away is that asphalt contributes quite a lot to worker exposure and may also contribute to urban air quality. And another thing is that personal exposure is a good measure for the worker exposure, but we also have to know what the specific sources emit. And that's why it's important to rethink some of the ways we're doing our measurements. And also think about what kind of exposure pathways is present and how relevant are they for the working environment. Yes, thank you. And I could not have done this without all the other cool people from all the institutes I'm a part of. So.
Thank you. Merv, it's very strange calling you Merv. Just gonna put that out there. So if anybody has any questions for Merv about asphalt and all the different experiments that she's just presented, put them in the chat or raise your hand, and we'll take them from there. But it's very interesting and very nicely this connection between lab and field, because often it's quite difficult to do lab experiments that actually represent the field measurements that you do.
Yeah.
So very nice to see how well that comes together.
Oh, thank you. Yeah, it's like there's of course some difficulties still, but like there's also other things that we have done, like that indicates that the main processes that we see might be related to the things we actually measure in the lab. So it's nice to see that at least like some of the mass spectrums look similar and so on, but this is still ongoing study, and some of the data here is quite fresh, so take it with a grain of salt.
I was wondering this enhancement that you see from the presence of NOx. So the NOx that you that is available, is that from the actual paving process, or is that from combustion, like from that process, or like, where is it coming from?
So like for the chamber studies I did, what we tried to do was just to do a simple system. So in the field, I have measured NOx, but I have not tested it in the lab with authentic samples. But what we see here is like from looking at like more simple samples in a way, which is not simple at all, because this was like 1 month of measurements, and like it's just doubled all the things you have to do. But it's quite interesting, and because a lot of studies actually show that NOx would reduce SOA formation by scavenging some of like the peroxide radicals, as I understand, and that will like kind of lead to less SOA formation. But we see that it's much more enhanced. The recycling of H radicals is much faster with the NOx settings that we have, at least. Yeah. So
And are those NOx settings atmospherically relevant, or are they very high in the chamber so you can like see what you're actually studying?
no, it's like 60 ppb. Did 150 and 60, which is relevant for the kind of like actually in the lower range. So we try to just be like, okay, we want to have some settings that resemble some of the road, or like where asphalt fumes are present, and those are the ones we work with. So you could always do different like levels of NOx as well, but it's like you go forever with all these experiments. It's
Yeah, no, I'm just in like often when you do lab experiments, you do them at very high concentrations
yeah.
compared to what you would have in the field. So it's interesting to hear. So there's also a question from Sachin.
Yeah.
It's interesting to know that benzene delayed the SOA formation.
Mhm.
What could be the reason for it?
Yeah. So what we think is that when you have the OH concentration, it's quite similar for the experiments. So we put in the same amount of H2O2 to then actually produce OH radicals. So we think that the OH might, like I said, the benzene will take some of the OH radicals, and in that sense, like make the naphthalene, which is like the primary one that makes the SOA, because we tried also to get benzene to make SOA, but it couldn't. We tried really high levels of oxidants and it didn't work. So we kind of had to have naphthalene present in order to get NOSWA. But one interesting thing is we want to still test a little bit more, is that in the longer run, the longer we run, it might occur that benzene might increase some of the SOA yield at the end, which is quite interesting, because when you have, for example, in the atmosphere, we do modeling, we do experiments and so on, we just assume we have only one precursor and that it doesn't co-react with other things. And that's the one we usually calculate the SOA yield from. But having this combined mixture might actually affect urban air quality even more than we thought. So that's still something we are looking into, and I look forward to you doing SOA on authentic samples as well. But yeah.
Very nice. Are there any more questions for Merv? It doesn't seem like it. Then I will say thank you to everybody who showed up. I'm sorry that we had to reschedule it for this week, but there were some technical issues last week that were out of our control. And thank you to our two speakers. Fantastic talks. Thank you for being available to give talks in December. It seems a lot of people are unavailable at this time. And I hope everybody has a great day.
Thank you. See you.
Bye.
Bye.